Good morning, Shem Stars. In previous presentation of Chapter 12, Biotechnology and its applications, we discussed biotechnological applications, that is, genetically engineered insulin and vaccines. In this presentation, we will discuss about gene therapy and molecular diagnosis. Gene therapy is a collection of methods that allows correction of gene defects diagnosed in a child or embryo. Genes are inserted into a person's cells and tissues to treat a disease. Correction of a genetic defect involves delivery of a normal gene into an individual or embryo to take over the function and compensate for the non functional gene. First gene therapy was given to a 4-year-old girl with adenosine deaminase, that is ADA, adenosine deaminase deficiency. ADA is caused due to the deletion of the gene for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation and enzyme replacement therapy, but they are not completely curable. This can be treated by gene therapy. In first step of gene therapy, lymphocytes from the blood of the patient are grown in a culture outside the body. A functional ADA cDNA using a retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the patient. As these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. If the gene isolated from bone marrow cells producing ADA is introduced into cells at early embryonic stages, it could be a permanent cure. Some other diseases that can be treated by gene therapy are hemophilia, cystic fibrosis, and Parkinson's disease. Another important medical application of biotechnology, which we are going to discuss, that is molecular diagnosis. It helps to solve the problem of early diagnosis and treatment of diseases. Using conventional methods of diagnosis, that is serum and urine analysis, early detection of diseases is not possible. To overcome this problem, some molecular diagnosis techniques were developed that provide early detection of diseases. These are, which we have already discussed in the previous chapter, PCR or polymerase chain reaction. It helps in early detection of disease or pathogens by the amplification of their nucleic acid. Low concentration of pathogens, for example, bacteria, viruses in the blood does not allow its detection. PCR can amplify nucleic acids in such pathogens even when their concentration is very low. PCR technique can be used for detecting HIV in suspected AIDS patients, genetic mutation in suspected cancer patients, and in identifying genetic disorders. Another important application of molecular diagnosis, that is rDNA technology, recombinant DNA technology. It is a modern molecular diagnostic technique. As we discussed in the previous presentation, a single stranded DNA or RNA tagged with a radioactive molecule called probe is allowed to hybridize to its complementary DNA in a clone of cells. The cells are then detected by autoradiography. The clone, having muted gene, will not appear on the photographic film because the probe will not have complementary with the muted gene. The next important application of molecular diagnosis, that is ELISA test, that is enzyme linked immunosorbent assay. It is based on the principle of antigen antibody interaction. Infection by pathogen can be detected by the presence of antigens. It may be protein or glycoprotein, or by the detecting the antibodies synthesized against the pathogen. So, children, this is all about biotechnological applications in medicine. I hope you must have understood. In the next presentation, we will discuss about transgenic animals and ethical issues, which will be the last topic of this chapter, chapter 12. Thank you, Shem Stars. That's all for today. You can drop your queries on Google Classroom. Take care.